Good morning to all and have a nice and blissful day. Welcome to my class and let's start today the most iconic double helix structure of the DNA. Though DNA was first identified long back in 1869, but due to technical limitations in isolating such a long chain of DNA, such a long polymer of DNA, explanation or elucidation of the structure of DNA remained elusive, means बहुत दिनों तक DNA का structure उलझा रहा, भ्रामक रहा. It was only in 1953, James Watson and Francis Crick proposed this iconic double helix model. Both Watson and Crick were research fellows and doing research in Cambridge University, England. The double helix model of Watson and Crick based on two lines of investigation. Number one, X-ray diffraction study done by Wilkins and Franklin, that is Maurice Wilkins and Rosalind Franklin. They suggested that DNA is spiral or helical in nature. They must professors of your King's College London. Another line of investigation was done by Arvin Chargaff, did the base composition study of DNA and suggested that the base pairing ratio of adenine and thymine as well as guanine and cytosine are constant and equals to 1. Arvin Chargaff was a famous professor of Columbia University, New York. Both Watson and Crick took the help of these two professors and proposed the famous and iconic double helix model. Clear? And got Nobel Prize. Watson and Crick and Wilkins got Nobel Prize in 1962. But Arvin Chargaff didn't get your Nobel Prize. Clear? He didn't get the Nobel Prize, even though the iconic double helix model would not have been possible without Chargaff. So because Chargaff didn't get your Nobel Prize, he is often called as unsung hero of DNA. Clear? Now, one of the hallmarks of the double helix model is base pairing between two polynucleotide chain. Very, very important question. What is the hallmark of double helix model? If question comes, then it is base pairing between two polynucleotide chain. The base pairing follows a very unique property of polynucleotide chains called as complementarity. That is, both the strands of DNA are complementary to each other. What does it mean? Means if base sequence of one polynucleotide chain or one DNA strand is known, then sequence of bases of another strand of DNA can be predicted. So if each strand of the parental DNA, synthesizes a new strand, then two daughter DNA molecules are produced from the parent DNA molecule, and both the daughter DNA molecules are similar to your parent DNA molecule. Clear? Both are similar to the parent DNA molecule. Because of this genetic implications, the double helix model, the structure of double helix model became very clear. Remember children, that is Chargaff contribution only for double stranded DNA, not for single stranded DNA. Clear children? Have a look and please write a brief note of this double helix model. And the diagram of the parental DNA molecule is given here. You see towards the board, and how each strand of the DNA, parental DNA, which is called template, parental DNA strand is called template, forming a new strand. and thus two identical daughter molecules are forming from one parent molecule. Clear? Here the complementarity property means so P is complementary to A, G is complementary to C. That's why two identical daughter molecules are formed from one parent DNA molecule. Now I'll be discussing some salient features or the vital, so to say, important features of the double helix. I'll be highlighting some of the important points that are given in NCERT. The reason is NCERT points are very, very important for NEET examination. DNA consists of two polynucleotide chains. The backbone of this polynucleotide chain is formed by sugar phosphate. You see, I have written that is sugar phosphate form the backbone of the double helix. Okay, now the nitrogenous bases, have a look. Nitrogenous bases, they face towards the inner side. Nitrogenous bases, they project inward. Whereas I have already discussed, in a single polynucleotide chain, the bases are towards outside. But in double helix model, bases, nitrogenous bases, they face or project inward. Very, very important. Second point will be there, both the strands of the DNA, they are anti-parallel, means having anti-parallel polarity. One is 5,3 and another one is 3,5. Third point you see. Now base pairing takes place between two polynucleotide strands. Purines with pyrimidines and pyrimidines with purine. A pairs with T and G pairs with C by double and triple hydrogen bond, by double hydrogen bond and triple hydrogen bond respectively. This type of pairing, that is purine with pyrimidine, generates approximately an equal distance between the two strands throughout the length of the double helix. Clear? I have written this thing. Now the two chains are coiled in a right-handed manner, that is right-handed fashion. You see both the strands are showing coiling towards the right side, right-handed helix they show, both the strand. And right-handed helix is shown by your Watson-Crick double helix model. And now this Watson-Crick double helix model is called BDNA. Watson and Crick DNA is called as BDNA as because it shows a right-handed helix. Now children will be studying later on, question will be getting, but left-handed helix is shown by ZDNA. So BDNA means Watson and Crick DNA. This term is used recently. Now it is being used, Watson and Crick DNA, as BDNA. Next point you see. Each spiral, you see, one spiral or one pitch, or you can say one turn of the helix includes a distance of about 3.4 nanometer. So one pitch or spiral includes a distance, approximately a distance of 3.4 nanometer, which roughly includes 10 base pairs. That's why distance between a base pair is 0.34 nanometer. How it comes? Each turn or each spiral having the distance 3.4 nanometer. Now it includes roughly 10 base pairs. So 3.4 divided by 10, it comes as, or it comes to 0.34 nanometer. I have written this, children. Plane of one base pair stacks over another base pair. Clear or not? Stacking takes place between one base pair and another base pair. So stacking of base pair takes place in the double helix model. One base pair stacks over the another base pair. In addition to the presence of this hydrogen bond, it provides stability to the double helix. Clear, children? All the points are given. You can go through this and write a brief note. This is the salient features or the vital features or important characteristic of the double helix model to be remembered for example. Finally, now we are discussing some of the important questions expected in NEET and board examination from this double helix structural model of DNA. Have a look. There are some important questions. Number of hydrogen bonds in ATCGA. You can easily understand because A pairs with T by double hydrogen bond and G with C triple hydrogen bond. So you can count the number of hydrogen bond. The answer will be 12. Okay, children. In double stranded DNA, charge of equivalency rule is applicable, but not in single stranded DNA. So one question is there, if A adenine is 10%, then what would be the amount of your guanine and cytosine? A 10%, then T also will be 10%. So 20% out of 100, 20% if is going, then 80% remains, and 80% will be equally divided by G and C. So 40% will be G and 40% will be C. Now a DNA segment is there and showing the proportion of adenine, thymine, guanine, cytosine. Here all the ratios are different. So question is asked whether it is double standard or single standard. Had it been a case of double standard, then the ratio of A and T would have been similar. G and C would have been similar, but here different ratios or percentage you are getting. Different percentage you are getting ATGC, so it is a single stranded DNA, can never be double standard. Now next, single turn of helix. Having your single turn of a double helix, 10 base pairing includes. The question is asked, how many bases are purine bases and how many pyrimidine bases? 10 base pairs means how many bases, children? 20 bases. Clear? That's why 10 will be your purine bases and 10 will be your pyrimidine bases. Now come to the next. Length of the DNA is measured. The formula is given, that is number of base pair into distance between two base pair. And there is number of base pair into 0.34 nanometer. Clear or not? Distance between a base pair. So the answer will come. Now I have given one question. If the DNA segment 10,000 base pair, then what is the length of the DNA? The total number of base pair 10,000 into 0.34 nanometer, children. Now you know this E. coli having the DNA segment, DNA content is 4.6 into 10 to the power 6. So if you multiply by your 0.34, the length of the DNA will come. Same thing now. If the DNA molecule has 2000 base pairs, please see 2000 base pair, तो कितना bases होगा और nucleotides होगा 4000 nucleotides होगा 2000 base pair में, and each nucleotide has got one base, one phosphate and one sugar molecule. Clear? So question is asked, how many sugar molecule will be there, how many phosphate molecule, and how many N-glycosidic linkages, all will be same. Clear or not? So 2000 base pairs mean 4000 nucleotides. Each nucleotide has got one base, one sugar, one phosphate. So answer of sugar molecule will be 4000. Same thing, phosphate will be also 4000, and N-glycosidic linkage is also 4000. Question number 7, double stranded DNA. There are 200 base pair. It has 10% adenine. That's why 10% of 400 is 40 for both adenine and thymine. The rest your 40%. 40% of 400 will be 160, 160. So that will be guanine and cytosine. Clear? Tm value, temperature of melting, is Tm value is directly proportional to the GC ratio. Clear or not? The Tm value is more in prokaryote because GC is more in prokaryote, whereas in eukaryote, adenine and thymine is more. So the Tm value is less in eukaryotes because GC less, AT more. बेस रेशियो एक चार्ज ऑफ इक्विवेलेंस रूल में बेस रेशियो एक क्वेश्चन आता है जो कि कांस्टेंट फॉर ए पर्टिकुलर टाइप ऑफ स्पीसीज A plus T by G plus C is equal to constant for a species. These are some important questions from this double helix chapter. Okay children, thank you.